Hi guys, David here with another science update. So today I want to talk about a really interesting paper that pertains to the idea of using carbon nanotubes to potentially regenerate damaged nerve tissue. And this paper is from researchers at the University of Hiroshima in Japan, and it's called Carbon Nanotube Yarns Induce Axonal Regeneration in Peripheral Nerve Defect. So we're talking about carbon nanotube yarns, and we'll talk about what that means in a second. Axonal regeneration means the ability to regrow axons, which are the long, thin part of nerves that allow a signal to be transduced from one part in the body to another part in the body. And when we're talking about peripheral nerves, we're talking about basically nerves that aren't part of your spinal cord. So carbon nanotubes are just another allotrope of carbon, which is to say another form of carbon. And carbon's a really interesting material because it comes in so many different forms for its pure elemental composition. And so these are just some of the different kind of molecular structures that carbon can assume. So this one is basically the pattern of atoms for diamond, whereas this here is the pattern of atoms for graphite. And then we have down here, this is a carbon nanotube, and you can see that basically it has this tube-like structure. That's essentially it. So the paper says an injured peripheral nerve can regenerate spontaneously or via a microsurgical suture. However, in long-term defects or severe injury, the regeneration capacity of the nerve is limited. And we know this, of course, is that in some cases, if you have a minor injury to a nerve, you can your body can basically heal itself. But if you have a major injury, then sometimes you just never recover that function. So although this is not the first time that carbon nanotubes have been explored for nerve regeneration, this is one of the only in vivo studies that's ever been conducted. And an in vivo study is where they're actually studying something in a living being. So let's take a look at the materials that they actually are using. So this image A is a cross section of the fiber. And what they have is on the outside, they have basically a silicon structure. So they have a silicon tube, and on the inside are the carbon nanotubes. So these are microscope images, and we're zooming in. So in section B, you can see now you're looking at the nanofibers themselves, or the carbon nanotubes, and you can see that they're nicely aligned. And then section C is the external surface of one of these carbon yarn fibers. So they zoom in even more. And then section D is what it looks like when you zoom in, and you can see the magnified external surface of the carbon yarn fibers. And so this is what the carbon nanotubes look like if you really zoom in. And so what they're going to do is they're going to take these bundles of carbon nanotubes that are in this hollow silicon tube, and they're going to basically put that in a place where the nerve has either been removed or damaged, and they're going to try to find out if the nerve will regrow along that carbon nanotube and restore function. So what they did is they did this study with rats, and they had 5 experimental groups, and there's a total of 35 rats. C0 was a control group, and it just had a hollow silicon tube, but with no carbon nanotubes. Then CN2 was the group that had the carbon nanotube yarn filling at a low density of 2% in the silicon tube. So they had some of the carbon nanotubes in that hollow tube. And then CN5 had a moderate density of 5%. And then CN10 had 10%. And CN35 had a very high density of carbon nanotubes at 35% filling the silicon bundle. And what they did then is they basically did surgery on these mice to remove some of the nerve tissue and then implant these carbon nanotube bundles inside the silicon tubes. And then they watched to see if the nerve function would if the nerve would regrow and the mice or the rats would regain their function. These are images after 8 weeks after surgery. You can see C0, so that's with no nanotubes. C2, that's 2% of the nanotubes. CN5 is 5%, etc. And what we're looking for here is, you can see in CN2, for instance, this white or this light colored tissue has actually regrown. And CN5, it's done the same thing. C0, you don't really see too much of that, and neither do you do it with CN35. So what's interesting is if you have too much of the nanotubes, it looks like it impedes the growth of the axon tissue, the nerve tissue. And if you have too little, it also doesn't really grow effectively. Here we see a light micrograph of the carbon nanotubes with a nerve tissue with staining, and you can see C0. It's interesting, you have tissue has grown, but it's not complete, and there's gaps in the axon. Whereas CN2, CN5, and CN10 all basically have a continuous filament. An axon has regrown along the axes of the nanotubes. And then CN35 is interesting. You just see, it looks like to me there was probably just too much material there, and there wasn't space for the nerve cell to actually regrow. So it looks like too little of the carbon nanotubes and too much of the carbon nanotubes was detrimental to growth. They also say, although we were able to demonstrate the effectiveness of CNT yarns in peripheral nerve regeneration, this study has several limitations. First, we used an artificial nerve model that combines a silicon tube and a carbon nanotube yarn, which is far from clinical use. In other words, these materials, they were pretty effective in this study, but the chances that these are going to be approved for use in humans anytime soon is probably low. And they say in conclusion that although CNT yarns require further development in terms of therapeutic effect and biocompatibility, the results of this study support the future clinical application of CNTs as an off-the-shelf material for artificial nerve conduits. So this is really cool. I think this is a very interesting study, and I've linked it down below. It is open access, so you should be able to access it no problem. And I hope that they can do some follow-up studies, and maybe in the future this can be of real use to people who have experienced kind of long-term nerve damage that doesn't regrow on its own. So the other article that I want to talk about today is called Bugs in Space, the battle to keep the ISS clean. And it says the International Space Station is teeming with bacteria, so how do you keep a space station clean? They say it turns out that if you take a partially closed built environment and fill it with humans for a couple of decades, you'll end up with a pretty interesting microbial community. This shouldn't be a surprise. In 1998, astronauts aboard the Russian space station Mir were curious to know about the microbes they were sharing their living space with. When they opened an inspection panel, they found large globules, right, because it's low gravity, so there's globules of murky water teeming with bacteria, fungi, and mites. Even more concerningly, they discovered organisms eating away at the rubberized seals around the windows, and bugs that excrete acid working their way through the electrical cablings. Can you imagine how terrifying that would be if you're in a spaceship and you realize all of a sudden that there are literally, teeming hordes of microbes actually destroying the spaceship itself? The ISS was launched that same year in 1998, and at the time it was absolutely pristine. And it's been going now for more than 22 years. It's been continuously occupied over that time by a stream of nearly 250 astronauts, and all of those humans have brought with them a complex assortment of microbes. So they've identified more than 55 species of bacteria on the ISS. They've also found fungi, molds, protozoa, and viruses. Interestingly, despite the lack of gravity, many of these species have adapted well to their surroundings. With only a small amount of gravity, many normal processes like convection and buoyancy are minimized, while others like liquid surface tension and capillary forces become more intense. While it's not entirely clear how these changes affect microbes, studies suggest that bacteria mutate quicker in space to adapt. So that's pretty horrible. So it goes on, how do astronauts clean the space station? For astronauts, Saturday is not a day of rest. They're up early, sticking to their strict cleaning schedule. We have to disinfect all the surfaces we touch every week, astronaut Thomas Pesquet said in a call with journalists early in the year. We also have a lot of measures on the space station that are similar to those you can find in hospitals or at airports that are designed to prevent the propagation of bacteria. And that's a good point. Can you imagine being sick in space? I don't know if that's happened. I wonder if anyone's had a cold in the space station or gotten the flu, God forbid. And to me, this brings up an interesting point just about the future of space travel. I think that we're going to have to be very, very careful when we do long-term space travel or missions that last months and years, that we can control the propagation of microbes in our atmosphere. I also know that human beings, since we're symbiotes with so many different bacteria and things like that, I think that sometimes you can cause health problems by being in too clean of an environment as well. So I'm not sure what that balance is. How do you get clean enough to avoid things like this murky globules of water teeming with mites and fungi and bacteria, which is pretty horrifying, especially if they start eating away at the windows of the spacecraft. And how do you avoid, on the other extreme, making things so sterile that you're actually making a human being sick? I'm not sure. Maybe the future is that if we're going to do long-term space travel, maybe we just need to have like very, very large spaceships which have intentional biospheres in them, like with trees and things like that, like coal gardens. And then we could actually maybe engineer an environment that has good microbes in it, and those good microbes can kind of prevent sort of just all these like staphylococcus bacteria and things like that from being like the main microbes that exist in the space station. Obviously that would take a lot of work, and I think we're pretty far from being able to make something that large, but maybe something along those lines is the future. Anyway, thanks so much for watching. This has been another science update. If you enjoyed the video, please like and hit subscribe, and I hope you'll join me again in the future soon. Bye.